are you facing the problems for kit settings in bioanalyzers or pathological laboratory? Here in this video, I will solve all your queries regarding for the kit setting. Hi guys, welcome to my YouTube channel. In this video, we will see how to set the chloride kit in every semi-bioanalyzer. Before proceeding for the chloride kit setting, I will show its clinical significance and why doctor refer for to diagnose the chloride test. The chloride is been tested to measure the amount of the chloride in our body or in our blood. It is the type of the electrolytes. Electrolytes are electrically charged minerals which helps to control the amount of fluids and balance the acid or base pH value in our body. It is often measured with other electrolytes to diagnose the kidney functions, heart failure, liver disease, high blood pressure, etc. The symptoms are like vomiting for a long time, diarrhea, fatigue, weakness, dehydration. The normal values for the chloride is about 98 to 107 millimoles per liter. Now we will proceed for the kit settings in semi-bioanalyzers by the manufacturer Perfexn. Before setting for this kit in the semi-bioanalyzer, we have to switch off the analyzer 10 minutes so that all the software program will be set in the analyzer. So you will see on the screen there is the menu options are there. So go for the test menu. Go further in the open channel. Now you have to proceed for the edit name to which we have to test. Here please note here I am working with the Perfexn manufacturer chloride mono kit. So on the screen you will see this. You have to go for the edit. Now go to the parameter. In the basic you will see these options are there. So we have to fit accordingly to the parameters given by the literature. So go to the backspace and enter the name of the test which we have to perform. So we are performing for the chloride. Now go for the blank. See the parameter given in this. So for the blanking it is given as the reagent. So enter for the reagent. For the units it is millimole per liter. By side button we have to press for the millimole per liter. Now further we are working with the reagents. The reagent volume, here you will see the reagent is 1000 microliter. So we have to enter for the 1000 microliter. For the sample requirement it is 10 microliter. This has been fed. And for the aspiration by the analyzer is 450. The factor will come after we do the calibration for the standard. Now further it is test mode, endpoint method. For the primary wavelength, here they have given 505 wavelength. So enter for the 505. For the temperature it requires the room temperature, so remain as the room temperature. For the delay time it is approximately 5 seconds. Now we have set all the programs accordingly to the literatures given by the Perfexn. Now save it and go to the return. Now go for the calibration, enter it, and we have to enter the concentration of the standard. You will see on this vial, because they have given here 100 millimole per liter. So we have to enter for the 100 millimole per liter. Here they have given. So save it and run for the test. Before proceeding for the test, I will tell the linearity of the chloride reagent by the Perfexn is 130 millimole per liter. Before proceeding for the test, I will tell the requirement for the chloride test. We require the reagent, chloride monovials. We require the 100 to 1000 variable pipettes, micropipettes, 5 to 50 micropipette. Here are the clean dry tips. The sample. I am working with the sample provided by the biograde, that is level 1. Distilled water, test tubes, tissue paper, etc. So now we will proceed for the chloride test. I have taken the vials of chloride. I label it as the blank, standard and taste. So to the blank we will not add anything. because we have to aspirate as a blend grid for the blend, and they have provided the reagent up to 1 ml over here, means 1000 microliter. You will see over here 1000 microliter. In each tube it has the 1000 microliter. So we have to add to it the concentration of the standard, that is 100 millimole per liter. So I adjusted the micropipette of 10 micro. Here I am adding the standard of the chloride, that is 100 millimole. Take the standard of 10 micro, wipe the excess. Now we have to add it to the standard labeled tube. We have to incubate for 10 minutes at room temperature. Taken the sample of the Biorate control level 1 as a sample of serum, 10 microliter. You will see, wipe the excess and mix in the labeled tube of the sample test. Mix well and incubate it for 10 minutes at room temperature. Here our 10 minutes have been over. So on the screen you will see aspirate for the distilled water. Now on the screen you will see here are the menus over there. So we have to press for the reagent blank over here, as we have to proceed for the aspiration for the reagent. Note the absorbance of the blank, that is 0.015. Now we have to press for the calibration to read the reading for the standard. We have to mix it first. Now we will aspirate for the standard of 100 millimole per liter. On the screen you will see the absorbance of the standard, that is 0.202 and the factor is 494.84. So we have to press for the enter, that is OK. Now we will proceed for the reading for the sample, as we have taken the Biorate control of level 1. So aspirate, test sample. On the screen you will see the reading of the chloride is 87.95 of the Biorate control of level 1. Here I am showing the control sheet of the Biorate control chloride reading is there. This is the target 92.3 and the target range is 81.2 to 103. So our reading is in the target range. As our result is in the target value, so here the reagents taken from the Perfexn, which is giving the best results with an authentic value. So here we have learned for the chloride test for its clinical significance, symptoms, kit settings in semi-bioanalyzer, procedure for the chloride, etc. So if you have any query, drop in the comment box, and if you like the video, please share and subscribe to my YouTube channel.